Hi friends, welcome back to the JK Biology, and today I will be described about the Maxam Gilbert DNA sequencing method, that is also called chemical degradation method of DNA sequencing. Okay, so what is the steps of the Maxam Gilbert DNA sequencing? That is here I have written already that in the first steps 5-end labeling. Okay, second step you see restriction endonucleotidations, and the third step is denaturation of the double stranded DNA. Okay, and the fourth step is the chemical degradation, then gel electrophoresis, and further autoradiography, and then sequencing determinations. Okay, so in the first step, how we end the file of the DNA, how we end label the 5-end. Okay, then this is the alkaline phosphatase enzymes in the double stranded DNA that will remove the 5 phosphates. Remove the only 5 phosphates, and also there is 5 phosphate that's become removed by the alkaline phosphatase, and so remaining of the 5-OH group. Okay, and also in that condition OH groups, because alkaline phosphate remove 5 phosphate of the ends. Okay, then polynucleotide kinase is an enzyme that add the phosphates. Why we require to remove the phosphates and again add the phosphate groups? Why? Because that is not radio labeled. That's become not radioactive. Okay, so first we should need to remove the 5 phosphates, and so we should need to add the radioactive phosphate, that is 32. Okay, that's emit radiations to detect or make the photographic film by the autoradiography. Okay, why we require? Also I will describe that's why we require this radioactive element, that is 5 phosphates. Okay, and then all the DNA when becomes phosphorylated by the radioactive phosphate, then restriction digestion. Why require restriction digestion? Because the length of the DNA becomes very large, that's become big, so we need to require short DNA sequences. And also in the gel electrophoresis, the large DNA cannot be separated easily. Okay, so it's need to require shorter length. Then we need to restriction digestion. When the restriction digestion takes place within the DNA sequences, then the 2 fragments will be obtained, either maybe 4 fragments or 3 fragments may be obtained in the DNA sequences. Okay, so suppose that there is the 2 fragments DNA sequences is obtained here, then what is need? The all is the radioactive labeled, then this is denatured, but this is taken separate forms. Okay, this is taken separate tubes for further sequencing, and this is taken into separate tubes. Okay, both, because we need to all the sequence of the DNA. Okay, then there is the restriction digestions that will take another reactions, and that will take another reactions. But after then we take only, suppose that this is the taking single sequences. I am taking showing only single sequences, not these sequences. Okay, then we carry to these sequences required denaturations of the DNA to be single stranded. Okay, because without single stranded DNA we cannot sequence. Okay, so that is denaturation steps that make the double stranded DNA sequences into the single stranded DNA. Okay, this is 3 OH, that is 5 phosphates. Also here in the another reaction mixtures or another process that will require denaturations, and that will use to the sequencing of the 5 ends. Okay, and this is required to the only 5 ends, so this is not required. Okay, and that is also not required in another reactions, so only this is required, this single stranded DNA. Further, then how we can determine these reactions? Okay, this step is end to the denaturation process. That steps cover 5-end labeling and restriction digestion and then denaturation. After then that's come chemical degradation methods. Okay, then what is the chemical degradation methods? Suppose that there is, we make the denaturation by the single stranded DNA, that is, then after denaturation to find single stranded DNA, suppose that this is the single stranded DNA sequence is here. Okay, 5 ends, 3 ends, 5 and 3 ends. That is phosphorylated with radioactive substance by the polynucleotide kinase you have known. Then these sequences is transferred to the 4 helicodes. That is the first helicode, second helicode, third and fourth. That is helicodes means separate tubes. Okay, that is separate tubes. First, second, third. Why? Because in the first tube we add dimethyl sulfates, piperidine. Okay, where already DNA sequences, single stranded DNA sequences, that is radio labeled, is added, and within this, that's the DMS and piperidine is added. Why? These degrade the nucleotide sequences. How? How these degrade the nucleotide sequences, DMS and piperidine? Okay, then suppose that this is the GTCA sequences. I am making a small chart of the DNA sequences. That is, there is the sugar bases. Okay, then the nucleotide is added in these forms by the glycosidic bond. What is that? Is G. You know that's purine and pyrimidine. Purine, adenine and guanine, but pyrimidine is cited in thymidine. Okay, then that is G and T, further C. For simple example, for understanding to you, that's how DMS, piperidine and different chemicals digest the DNA sequences. Okay, and further that is A. Okay, then dimethyl sulfate specifically recognize those base pairs where G is available in the DNA sequences in this tube. Okay, where G is available in these sequences. So you know, you are seeing here that there is 2G, 1 and 2. That will be methylated. That will be methylated. What happened after methylation? After methylation, the glycosidic bond will be break. Okay, the glycosidic bond will be break. Then this will be removed. Point out, this will be removed. And again, there is also G. This will be methylated and the glycosidic bond will be removed. Then what happens? Piperidine is the substance, that is the chemical that recognize modified bases, that recognize those bases where any nucleotides glycosidic bonds have been break and release the nucleotides. Piperidine functions. So piperidine, you will see here that piperidine is added in all the tubes. Why? Because all the tubes contain different, that is DMS, formic acid, hydrazine and so 1.5 millimolar NaCl. This is different. That's on the basis of chemicals, the methylation would occur on specific base sequences. DMS methylates only G and formic acid plus DMS methylates both purine, both, that is guanine and adenine. Guanine and also where adenine is available. Methylates both. When methylates, the glycosidic bond will be breaks. Okay. Also, hydrazine methylates on cytidine and thymidine. When thymine and cytidine is methylated, the glycosidic bond will be removed. What happens? When the glycosidic bond will be removed of the bases, then that is removed. In that conditions, only G is methylates and removed. Then piperidine recognize the phosphodiester bonds and break. What happens? You will see here that this base will be removed. Okay, after digestion of the phosphodiester bonds by the piperidine. Then G is totally removed. The sequence only G is only obtained here, one basis sequences. Phosphate is only leaving phosphate. G is removed, only leaving phosphate. You will see here, that's only phosphate at the 5 end is here removed. And what is the main aim of the adding to the radioactive substances? Why? Because after removal of the single phosphate, this will be remain. This will be remain and in that aliquots. Okay, this will remain in that aliquots, and these sequences will also run in the gel electrophoresis. Okay, so if we not added radioactive substances to the 5' ends, then what happens? These sequences will be also looking, and these sequences single nucleotide will be also looking. So there would be more confusion by mixing, overlapping. So when this end is radio labeled, that will make photographic film on the radioautography in the gel electrophoresis. Point out, in the gel electrophoresis, the only 5 ends show the photographic film, because emit the radiations, radioactive substances emit the radiations, and that will visible on the gel electrophoresis. Okay, and these sequences, remaining sequences would be not visible. Points out, this would be not visible. Okay, so the only one bands would be obtained, because there is G. In some cases, there is a lot of DNA sequences in this tube, lot of DNA sequences. So probably that will be hydrogen. Simultaneous reaction will take place. I have described about the Sanger sequencing methods. Lot of DNA sequences is available. In some DNA sequences only G is removed by methylation. In some sequences also in the middle, that is 1, 2nd, 3rd, 4th, 5 and 6. 6th position of the G will be methylated simultaneously. In some DNA sequences, point out that G is released of the 1st position, and of the 6th position also G would be released. So 2 types of the sequences would be obtained in the G. Here 2 types. That is 2 of the length. That is 1 length, and 1, 2, 3, 4, 5, 6 length. That is 6 length. You can see here in the gel electrophoresis there is the 6 length and 1 length of the G residue. The gel is prepared in those way that single nucleotide difference can also easily identification would be occur. Okay, easily identified, easily separates in the gel. Okay, so one nucleotide differences, that is G, and there is in the 6 positions. Okay, in the 6 position, you will see here that this is the 6th position. Okay, because there is the 6, the glycosidic bond will be released and G is removed. Okay, in this case, but in some cases, in another tube, DMS plus formic acid plus piperidine, after adding both purine bases would be methylated and removed by the piperidine, as I have described before this. Okay, that's removed in this middle sequences. So there is both sequences would be obtained. You point out that A sequences and G sequences both will be methylated and both will be removed, and both type of sequences would be obtained in the gel. That is G sequences as well as also A sequences. So G sequences breakage would be 2, and A sequences you will look here, that's only one of A sequences is here. So one sequence would be obtained, that is 1, 2, 3 and 4th. 1, 2, 3 and 4th. That is A sequences will be obtained. 1, 2, 3 and 4th A sequences, and this A sequences is not present in the G sequences. Point out, because there is not methylation at the A, because only DMS is available. Point out, in this aliquots, in these tubes, only DMS is available, so it will methylate only G residue, not A residue. So there is not methylation, not removal of the glycosidic bond, and not removal of the phosphodiester bonds. Okay, so there is not A bands, only G bands 1 and 6. And in this aliquots, you know that both methylation would be occur, both glycosidic bond will be break, and both phosphodiester bond will be released, and the base sequences would be the release. So simultaneously more of the DNA sequences is available here. I am showing only 4, not 4, a lot of DNA sequences. Okay. Then the band would be 1 of A and 2 of G, that is same 6 and that is 1 of the G. Okay. So this was the second cases. In the third aliquots or the tubes, we will take the pyrimidine residue. The pyrimidine, okay, cytidine, thymidine. And in these conditions, hydrazine is added, not formic acid DMS. Hydrazine added, hydrazine methylates both of the bases, that is pyrimidines, cytidine plus thymine. So you will see here that there is 1, 2, 3 thymine. Points out. This is 1, 2 and 3 thymine. And what is cytosines? 1 and 2 and 3 also 3 cytosines. So points out on the basis of these sequences, there would be 3 fragments of the cytosine and 3 fragments of the thymine. Points out. This in the first cases, that is 1, 2, 3. 1, 2, 3 cytosine, 3 bases pair longs. Okay. There is cytosine, 3 bases pair longs. Okay. 1, 2, 3 here. You will take here 3 bases long. Okay. That is cytosine. And again 1, 2, 3, 4, 5. That is 1, 2, 3, 4, 5. 5 bases pair long. Cytosine is also obtained here. 5 bases pair long also obtained. Okay. 5 bases also obtained. And there is, okay, 5 base pair also obtained. That is 3 base pair. And that would be not here. Point out. That is 1, 2, 3. 1, 2 and 3. In the 3 positions, it will be available. Okay. And again, T, there is T residue. 1, 2. So there is 2. T of 2 residue would be here, and that is 2 residue of the DNA sequence would be here. Okay. 2 residue. That is 2 residue. And this is 3 residue. That is cytosine. 3 residue. Okay. Cytosine. And again, after then, 1, 2, 3, 4 and 5. 1, 2, 3, 4 and 5. That is cytosine. There is cytosine is available after then cutting, after methylations, after removal. 1, 2, 3, 4, 5. 1, 2, 3, 4, 5. Okay, that is in the 5 positions. And again, that is this was 1, 2, 3, 4, 5 and 6, 7. In the 7th position, T. In the 7th position, T. 1, 2, 3, 4, 5, 6, 7. 7, that is T. Okay, that is T. Again, in the 8th position, that is T. In the 8th position, that is T. Because T is available here, T and cytidine. Okay. Then cytosine. Again cytosine there is modifications. So 1, 2, 3, 4, 5, 6, 7. 1, 2, 3, 4, 5, 6, 7, 8 and 9. 9 base pairs. So in the 9th position, that will be C. The length of that. So this is the sequences in the gel of both cytidine and thymine. Count that. What is cytosine number? Cytosine, thymine. That is 3 cytosines, 1, 2, 3 and 3 thymine, that is 1, 2, 3. So the number of bands would be 1, 2, 3, 1, 2, 3, 6. 6 bands. Combining of these 6 bands would be obtained. Okay. So this was the combination of cytidine and thymidine. In the case of cytosines, you can easily understand that there is in the positions of the 9th, there is C, there is C. Okay. And in that positions also, you will see that's 1, 2, 3. That is in the 3 positions. Okay. In the 3 positions, there is this, and 1, 2, 3, 4, 5. In the 5 position, that is here. C. The bands. So that was showing the number of nucleotides. That was showing the number of nucleotides after digestion in the separate gel. Okay. So you will see that this is the band of what? G. Okay. of, this is the band of G, G plus A and C plus T, and this is band of C. Okay. So this was clarification, but you know that shorter the nucleotides, longer the traveling. Longer travel on the gel if the shorter the nucleotides. So this is the one, number of nucleotides is 1. So there is longer traveling, and there is also a same, so in the first position. And the second position you will see here that on the basis of length there is traveling of the nucleotides in the gel electrophoresis. So there is what? 5 positions. G, there is 5 and that is 3. That is 5 and that is 3. So this is 5A, sorry, G, T, on the basis of U, this you can say that 5, and G, T, C, A, C, G, T, T and C. You can make the sequences, okay? G, T, C, A, C, G, T, T, C. That is the sequences from bottom to top. Okay, from that is bottom 5 end and top 3 ends. So in this Maxam Gilbert methods, you will find the differences from the Sanger and Maxam, that these will be obtained both overlapping fragments, that is in the C plus T and G plus A, that is matching. Okay, that is matching. C plus T and C, G plus A and G. Matching sequences would be obtained. This is the Maxam Gilbert of the DNA sequencing method in the gel. But you have seen in the Sanger method of the sequencing, there is not any overlapping of the bands in the gel electrophoresis. There is not any overlappings. Okay. So questions can be asked by drawing the gels, that what is the gel following the Sanger DNA sequencing method and what is the gel following the Maxam Gilbert sequencing methods. So you may be confused, but by showing this, that this is the overlapping sequences C plus T and G plus A, by this way you can match, and in the Sanger sequencing methods only any one bands cannot match. Okay, there is also 4 helicodes and there is also 4 lane of the gels, but there is not any matching. There is not any matching because there is not same length of the DNA sequences. So this is the differences of the Maxam Gilbert method of DNA sequencing on the gel and also of the Sanger DNA sequencing method of the gel. That would be the differences. So this was the method of the Maxam Gilbert method of DNA sequencing, and after getting bands only 5 ends is available. So after autoradiography, that will give the photographic films and the sequence would be the determined. So hope this lecture would be beneficial for you. If you like this lecture, then please click on subscribe button and also share for your friends. Thank you.